So, well, hi everyone, and we are here back as every Friday. So this Friday we have Maria Virginia Sanchez Puerta from Mendoza, Argentina. She's going to present about the tribe Hyoscyameae. We're very excited to hear about that. Well, she's from Bahia Blanca, Argentina, where she obtained her degree in biology. Then she moved to the US for her PhD in cell biology and molecular genetics of the University of Maryland with a Fulbright grant. And she focused there on the evolution of apophytes and dinoflagellates. And then she did a postdoc at Indiana University with Chef Palmer, and also but working on evolution of plants, especially on horizontal gene transfer. So by 2008, she moved back to Argentina, to Mendoza, where she started her own group, working mostly focused on evolutionary genomics of plants with emphasis in horizontal gene transfer and also mitochondrial genome recombination. So Virginia is a researcher of CONICET in Argentina and also a professor at the National University of Cuyo. And as I said before, she will present today about the tribe Hyoscyameae, somatic hybrids, phylogenetics, and horizontal gene transfer. So thank you very much, Virginia. If you want to share your screen.
Okay. Am I sharing it?
Yep, that's good.
Okay, excellent. Thanks, Rocio. And thanks all three of you for inviting me. And I'm very excited, excited to join this group during the lockdown year. So everything looks good? May I start?
Yes, you can start. It's perfect.
Excellent. So the title of the talk, the tribe Hyoscyameae, somatic hybrids, phylogenetics, and horizontal gene transfer, is a little bit of a potpourri of several topics that we've been working in the last few years in our lab, and that again like surround this tribe of Solanaceae. And the main focus of our lab is plant mitochondria, and this is because plant mitochondria are very particular, have very distinguishable characteristics such as their genomes. They are large and they are highly variable across angiosperms, with the smallest one being 66,000 base pairs in a hemiparasitic plant to 12 million base pairs in a species of Silene. And these genomes, even though they derive from endosymbiotic bacteria, they are not a single molecule, circular molecule. Instead, the genome consists of a population of circular or highly branched linear molecules, as can be seen here on the right. So the master circle that is normally published in the articles is just like an artifact of assembly, as the master circle has never been observed, only circular of subgenomic molecules. So even though they have a wide range of sizes, they all present between 30 and 40 per-encoding genes. That means that intergenic regions are extremely large, and they can be up to 98% of the mitochondrial genome. The origin of these intergenic regions is one unanswered question. Part of the intergenic regions consist of multiple repeats, can be short or large repeats. But then other parts, we still don't know the origin of those sequences, as the turnover of the intergenic sequences is very rapid. In contrast to the mutation rate, which is very low, is the lowest of the three genomes of a plant. And also the genomes can be found in different rearrangements, even within the same plant, because plant mitochondrial genomes undergo frequent recombinations, and we're going to talk a little bit about that. And also because plant mitochondria go through rounds of fusion and fission, as has been shown here below by Shihan. So at a certain point, we can see a mitochondrion that carries no genome, or that carries several copies of the whole mitochondrial genome. And another characteristic that we are very interested in is that plant mitochondria are prone to horizontal gene transfer. So moving to that topic a little bit, among all cases of horizontal gene transfer in plant mitochondria, the COX-1 intron, which is an intron found in the mitochondrial gene, COX-1 is the most frequently transferred region among plant mitochondria, with hundreds and probably thousands of cases across angiosperms. And so this intron was first acquired from fungi, but then it has been transferred among plants for the rest of the evolution. And the reason of the sporadic distribution of this intron among angiosperms, so it's due to multiple transfers given by the fact that the intron contains an open reading frame, which encodes an endonuclease. And so an intron homing mechanism has been proposed to explain why this region is so mobile and is the most highly transferred region. So briefly, the intron homing region, the intron homing mechanism states that the endonuclease is capable of, side endonuclease, which recognizes the intron-less copy of the COX-1 gene in the recipient plant, for example, and cuts the two strands of the DNA. And this begins the exonucleatic activity of the cell, which in turn calls for the repair mechanisms. In this case, the mechanism to repair the double strand break could be double holiday junction or synthesis-dependent strand annealing, which by using the intron-containing template to repair the intron-less copy, that was the native copy of the plant, then now the native copy acquires the intron. So this is the way that the intron can be propagated. Across plants. And oops, sorry. The reason why the intron is so clearly the result of horizontal gene transfer is for the phylogenetic incongruence that we observe between the phylogeny of the plants as we know them, and then the phylogeny of the COX-1 intron, and all the lines that you see that are not parallel indicate cases of horizontal gene transfer. Here, all the taxa in red contain the intron, and the taxa in blue lack the intron. So overall, in all these studies that have been published, more than 100 transfers of the COX-1 intron have been described, but we can speculate that thousands of transfers occur along angiosperm evolution. And in a study in 2008, what we found is that the intrafamilial transfers were more frequent than expected by chance. So that is that the intron is more commonly transferred within members of the same angiosperm family. We don't know why this pattern occurs, but it could be because family members may share the same vectors, which could be responsible for transferring this DNA, or could be due to illegitimate recombination between members of the same angiosperm family. So at that time, we decided to study a large family that have not been studied before, which is the family Solanaceae, and we did this in collaboration with Lynn Boss, Dick Olmsted, and Jeff Palmer. We examined 426 species of Solanaceae that belong to 70 genera, as we can see here on the right. And everything in black lacked the intron, and the three red lineages indicate 3 cases of intron acquisition. These seem to be independent intron acquisitions. And we see here below, Brunfelsia hamaicensis is the only species of Brunfelsia that we studied that contains the intron, and this intron is very different from that from the other Solanaceae, and so we assume that they acquire the intron in a different, from a separate donor. And then the intron in Mandragora and the tribe Hyoscyameae are very similar. They are sister in the intron phylogeny. So we assume that that is the result of an intrafamilial transfer of the intron. Then we wish to understand how many transfers occur in the evolution of the tribe Hyoscyameae, and we found that the relationships among species of the tribe were not clear, and so we decided to make an effort to understand the relationships within this tribe and understand how many transfers can explain the sporadic presence of the intron in this tribe. So going back in time a little bit, the tribe Hyoscyameae consists of 8 genera: Atropa, Atropanthe, Physoclina, Przewalska, Scopolia, Hyoscyamus, Archihyoscyamus, and Anisodus. And so in 2006, the group of Dick Olmsted found a retrotransposon in the gene in one of the copies of the gene WOXY in Atropa. And so that revealed that this plant is polyploid and is the basal lineage of the tribe. And this was confirmed later on in this and other studies too. In 2008, again by Dick Olmsted, where in a phylogeny of the Solanaceae based in 2 plastid regions, we can see that Atropa is an early divergent lineage within the tribe Hyoscyameae. And then we see many nodes with low bootstrap support. And we find one high support here for the Hyoscyameae, and another one here that links Physoclina with Scopolia and Przewalska, forming a monophyletic group. After that, 2 and collaborators in 2010 increased the number of plastid markers to 6 and an alignment of 8,000 base pairs, and found a very similar phylogeny where Atropa was the early divergent genus. And then again, we find these 2 nodes here that are very with low bootstrap support, making the relationships unresolved, and the 3 genera, Scopolia, Przewalska, and Physoclina, and again, are recovered in a monophyletic group. We increased again the number of variable plastid regions in 2014 to 10 plastid regions and an alignment of almost 12,000 base pairs, and find pretty much same result in which Atropa is the early divergent lineage, and then we find these 2 nodes in green with very low bootstrap support, which means that we are not sure about the position of the affiliations of Atropanthe, Anisodus, and Hyoscyamus within the tribe. And what we see here in red are the taxa of Hyoscyameae that contain the intron, and in blue, those that do not contain the intron. And so only recently, we were able to produce a phylogeny based on 10 complete chloroplast genomes of this tribe that became available in the last months and 2 years. And what we found now is a very well resolved relationships of the tribe where Atropa is the early divergent lineage. And then we find 2 clades, one formed by Hyoscyamus niger, that's the only chloroplast available, and 2 species of Anisodus. And then on the other clade, we found Atropanthe sinensis as sister to the 3 genera Przewalska, Physoclina, and Scopolia. So based on this phylogeny, we can infer 2 COX-1 intron transfers that took place in the evolution of the tribe, 1 intron acquisition in the ancestor of all the Hyoscyamus, as all the species we studied contain the intron, and another one in the ancestor of Przewalska, Physoclina, and Scopolia. Given that these introns are identical to each other, so they are likely have been transferred from one to the other, probably from this clade to Hyoscyamus, then this would be an intratribal transfer. And going back to the Solanaceae phylogeny, then there are 4 COX-1 intron transfers that took place, 1 in Brunfelsia and the other 3 in Mandragora, in Scopolia, Przewalska, and Physoclina, and in Hyoscyamus, there would be 3 cases of intrafamilial transfers, as we've been observing before this study. So Then going back to more general patterns of horizontal gene transfer in plant mitochondria, the cases described are the highest in numbers across all eukaryotes, of course not prokaryotes, but among eukaryotes, plant mitochondria, HET is the most frequent. And when we see the characteristic of all the foreign DNA found within plant mitochondria, they share three characteristics. One is they correspond almost always to mitochondrial sequences. They are donated almost exclusively from other plants, and they are transferred as DNA and not as messenger RNA. Then if we think about the evolutionary impact of these foreign DNAs, well, in general, foreign sequences are non-functional, and they may have an impact in the evolution because mitochondrial GT increases the genetic diversity as a result of recombination between the native and the foreign gene copies. And if you remember that the substitution rate in plant mitochondria was too low, this increase in genetic diversity may have a interesting impact in the evolution and the diversity of proteins in plants. So taking into account the patterns that we observe in plant mitochondria, a few years ago, Rice and collaborator presented a model named the mitochondrial fusion compatibility model to explain plant mitochondria HET, in which the donor plants would transfer complete mitochondria to the recipient plant. The foreign and native mitochondria will fuse, and their genomes will recombine. In this model, then we can the mitochondrial fusion, this part of the process may be restricted to phylogenetically related taxa due to incompatibilities in the fusion mechanism. So for example, plants are in intimate contact with fungi, but fungal mitochondria are likely not being able to fuse with the recipient plant mitochondria. So their fungal sequences will not enter. Same with bacteria that live with plants, bacteria will not fuse with the plant mitochondria. So this leaves only plant to plant, mitochondria to mitochondria transfers, and that's what we observe. So once the two mitochondria fuse, then the intergenomic recombination that takes place within the recipient mitochondria is not well understood, and that's another aspect that we are interested in. Before moving on to the next slide, so the most speculative part of this model is how is this whole mitochondria transferred to the recipient plant, and that was explained by speculative mechanisms such as insects that could move mitochondria around, or even fungi can be the vectors, or through illegitimate pollination, in which the pollen transfers the whole mitochondria from one plant to the other. So now moving on to genomic recombination in plant mitochondria. So not much is known. Most of our current knowledge comes from Arabidopsis mutants. And what we learned is that recombination in plant mitochondria is under nuclear control, and the factors are very similar to those in bacteria. And when the Arabidopsis mutants were analyzed, what we observe are mitochondrial rearrangements as a result of homologous recombinations and illegitimate microhomology mediated recombination. So every time a recombination takes place, it means that the process started with a double strand break, as we can see here. So for several reasons in plant mitochondria, a double strand break can occur. And this double strand break needs to be repaired. Very rarely, the repair takes place through A, which is a non-homologous mechanism. We only observe that in the mutants, in which the factors that suppress the illegitimate recombination process are mutated, and so they are not capable to do their work. But the rest of the time, the double strand break is repaired through one of the three described homologous recombination pathways here is C, D, and E, which is the double Holiday junction, the synthesis-dependent strand annealing, and the break-induced replication. These three homologous recombination mechanisms were described in other eukaryotes in the nuclear genome. And so it's not clear if the three pathways may take place in plant mitochondria, or if the factors that control these pathways are actually present in plant mitochondria, and these could exist. So this is all work that is being done by us and other labs, of course. So one other thing that you need to understand to move on to our work is that if this double strand break is repaired by an identical copy, let's say we have 2 copies of the whole mitochondrial genome present, and the double strand break is repaired by the other copy, then the break is perfectly repaired and there's no rearrangement. But if you remember, plant mitochondria have many repeats. So it's very common that the double strand break is repaired using a template, which is a repeated sequences and not the same region in another copy of the genome. So every time the double strand break is repaired with the repeated sequences, then the result is a genome rearrangement. But we don't know much about that. So our experimental model are somatic hybrids, because they represent an extraordinary system to study the mitogenome recombination that takes place normally in mitochondria, but also that takes place after a transfer event, like in the model that we mentioned. Solanaceae is the premier system for protoplast fusions and somatic hybrid production. So we focus on this family to choose the taxa for the somatic hybrid production. And so on the left, we have the scheme of the protoplast from species 1. So this would be the wild type species. And we, in collaboration with Mikhailo Zubko from Manchester Metropolitan University, we were able to analyze somatic hybrids from combinations of Hyoscyameae niger, Phycocline orientalis, Scopolia carniolica, and Isolus tanguticus, and some species of Solanum, at least to try to produce these hybrids. And then we have the protoplast from species 2, which is always the same, is tobacco, an albino Nicotiana tabacum, which is a nuclear mutant incapable of producing chlorophyll. This is very useful in the selection process of the fused cells because tobacco will grow very easily in vitro and not the wild parents, but if the cells that are growing very happily are green, then are probably the result of protoplast fusion. So we perform protoplast fusions through a chemical treatment. And then, as we can see in the pictures here, after a few days, the protoplast, the fused protoplast, grew a new cell wall and cell division started, and Kali was formed after a few days and weeks. And then later on, they start to become green, and we chose the green colonies, the green Kali. From these and other studies, we know that in a somatic hybrid, the mitochondria fused and the mitochondrial genome recombines pretty quickly. So the result of a somatic hybrid is normally a chimeric mitochondria, while plastids do not fuse and they sort out quickly. So normally a somatic hybrid has either one type of plastid or the other. In our case, it would always be the green plastid. And the nuclear genome can have different fates, and that depends a lot on our strategy. So for example, after plant regeneration and transferring to soil, in some cases, we did multiple back crosses with a wild type tobacco. And so we ended up with a nuclear genome that is fully from tobacco, but the cytoplasm, that is the chloroplast, were from the other parent, and the mitochondria was chimeric. So in that case, the somatic hybrid would be called cytoplasmic hybrid or cybrid. And we studied both somatic hybrids and cybrids. So this is one combination of parents that we studied. We have two of these hybrids that we fully examined. So in this case, we use an albino tobacco and a wild type plant of Hyoscyameae niger, and after protoplast fusion and several back crosses, we ended up with this hybrid that we see here that looks a lot like tobacco, but their mitochondria is chimeric. The plastids are from Hyoscyameae niger, and well, like I said, and the nuclear genome is from tobacco. But we were interested in the mitochondria and tried to understand the recombination events that took place when the two mitochondria fuse. And because we had two different mitochondria, then we were able to see the intergenomic recombination events. The intragenomic ones are very hard to examine. So first, let's compare the parental mitochondrial genomes. Tobacco has a mitochondrial genome of about 400,000 base pairs, and Hyoscyameae niger 500,000 base pairs. And even though their sizes are very similar, they only share less than half of their genomes are similar. That is, 200,000 base pairs represent homologous sequences, and they are interspersed across the whole genome. They both have 37 protein mitochondrial genes, 3 ribosomal RNAs, and 22 tRNAs. So at the time of fusion, we know that the mitochondrial genome of tobacco got together with the mitochondrial genome of Hyoscyameae niger. Well, here we distinguish the unique regions, the amount of unique regions from one parent, the other, and the homologous sequences. And after, all the mitosis, the plant regeneration, and the back crosses, so there was enough time and a few years passed, we finally sequenced the mitochondrial genome of the hybrid plant. And of the original sequences present in the mitochondrial population of that fused cell, what we find is that the unique regions from tobacco are retained. Almost all the unique regions from Hyoscyameae niger are retained, but the homologous sequences were retained only one of the copies, because the homologous sequences, once in the hybrid, become some sort of repeated sequences within the hybrid. So only one copy of each repeat in general was retained. So we concluded here that the homologous sequences, which are non-identical repeats in the hybrid, are preferentially lost, as only half of the homologous sequences were present in the hybrid. The unique regions from each parent are mostly retained. 90% of the unique sequences are found in this hybrid. And all these occur through multiple recombination events. So let's look at those recombination events. Like I said, these unique and homologous regions are really interspersed between each other, both in the parent, and here, this is the hybrid genome. And so what we see here is the line is the genome. It's only part of the genome of the hybrid. And we see here in blue, this sequence is derived from Hyoscyameae niger, this one from tobacco, the next one from Hyoscyameae niger, this one from tobacco, and so forth. So every time we see a blue next to a red rectangle, that means a recombination event took place right here. So overall, we can count, well, I'll tell you the number later, but more than 30 recombination events between the parental genomes. This is a zoom of the genome, just so you can understand how we were able to recognize the recombination event. So this is the hybrid genome, and we are focusing at this boundary here between the blue and the orange rectangle. And these are the regions of the parents that are likely, that were involved in this recombination. So here we have a unique region from Hyoscyameae niger, which the part that is overlapping with the region of tobacco means that that's a region of homology. So that is a non-identical repeat that was found initially in the hybrid. And through this repeat, which is 3,000 base pairs long, the recombination that we observed in the hybrid took place. So we did this examination for each recombination site, and we found that most or all of them, all of the recombination sites took place in places where there was a non-identical repeat, that is between homologous regions of each parent. That indicates us that the events were the result of homologous recombination and not of illegitimate recombination. So overall, the two hybrids that we analyzed, we found 37 and 53 homologous recombination events. And from the three mechanisms that that I showed you before, and I explained a little bit later, the break-induced replication is the most common. We also found one case of non-homologous recombination, which is rare, but it was it occurred in a microhomology region of 7 base pairs that were identical between the two parents, but that's not long enough to elicit homologous recombination. So let's go back to the three mechanisms of homologous recombination that may take place after a double-strand break. And here, the double-strand break repair or double Holian junction pathway leads to a crossover, which is the most familiar one. SDSA will lead to one of the parents and the other one with gene conversion, that is a stretch from the other parent, and break-induced replication, which is a different one because only one of the ends of the double-strand break participates in the recombination. The other end does not participate. And then so we end up with one parental copy, and the other copy, the one that suffered the double-strand break, will be repaired by using the other parent copy. And that means that we'll have half of one parent and half of the other parent. So it looks like a crossover in which we have only one of the products of crossing over. So it's non-reciprocal crossover. So here, what we see in our data, so if we look at this hybrid, we never find, well, we rarely find the two products of crossover. We only find one and one copy, intact copy of the parent, which is indicative of break-induced replication. And we only rarely found cases like this in which SDSA took place. So because of the result of the recombination events is that we inferred that the break-induced replication pathway took place more frequently in the hybrids. So what about the gene content? At the time of fusion, we have the gene content from both parents, that is the 37 protein-coding genes, the 3 ribosomal RNAs, and the tRNAs. But after some time passed by, we found that several copies were lost. Some of them recombined to each other, and some of them were kept both copies. So in general, what we see is that a single allele, a single copy of each gene is retained. And this could be because coexistence of different alleles, the same gene, may be deleterious. In our hybrid, what we observed is that most genes came from tobacco and only a few were chimeric, and only 3 cases in which both copies were retained. And this was observed, like I said, in the 2 hybrids that we analyzed. Then we analyzed a different hybrid, a somatic hybrid, because the nuclear genome may be the combination of both parents still, but the mitochondrial genome, which is the focus of our research, is chimeric. So here what we did is a proplex fusion between albino tobacco and Physocline orientalis. where we have these cells divided and the plant, the somatic hybrid plant, growing in vitro. So after 5 years of vegetative propagation of this plant, we studied the mitochondrial genome. And so, like we mentioned before here, the tobacco genome, which is 400,000 base pairs, is combined with the mitochondrial genome of Hysocyameae orientalis, which is 580,000 base pairs. And what we observed is that the genome of this hybrid is smaller than the sum of the parental genomes. That is, part of the genomes were lost. And what we observe is that homologous regions are preferentially lost and unique regions are preferentially retained, similar to what we observe in the other hybrids. A single allele is retained for most genes. And when we studied recombination events, we found 99 homologous recombination events, and most of them were the result of break-induced replication. And we also identified 3 non-homologous recombination events. So very similar patterns as in the independent hybrids that we analyzed. So in conclusions, the break-induced replication pathway seems to be the most frequent mechanism of homologous recombination in plant mitochondria. And here is responsible for the loss of the homologous parental regions. Somatic hybrids preferentially retain unique regions from each parent, maybe because they cannot undergo homologous recombination, which is the most common form of recombination. And a single allele of each gene is kept in somatic hybrids. It's likely that coexistence of different alleles may be deleterious because they affect global gene expression, but we don't really understand why is this affecting the expression. But the truth is that in 3 cases, we always found a single allele. We think that sequencing additional somatic hybrids may help to elucidate the universality of the patterns of recombinations and changes in the mitochondrial gene content. So we continue to study somatic hybrids to understand the patterns of recombination and fates of the mitochondrial genome of the hybrid. And so what do we expect to see in a horizontal gene transfer event in which the donor and recipient mitochondrial DNA interact as proposed by the model? So foreign sequences are likely incorporated into the recipient mitochondrial DNA by homologous recombination. That means that non-homologous foreign sequences will be incorporated less efficiently. And it is expected to find a single functional allele of each gene in the recipient mitochondrial DNA, either the native, the foreign, or a chimeric form, but it would be rare to find 2 full-length copies as apparently plant mitochondria can, in less than a few generations, can get rid of one of the alleles so easily. And finally, an unexpected negative result, and going back to the intron homing, another reason for choosing this Solanaceae and these parents in particular is that both Hyoscyameae and Hysocyameae contain the COX-1 intron, and tobacco lacks the COX-1 intron. And as I mentioned before, this is the intron homing is a proposed mechanism of intron propagation, but has never been shown, not even that the endonuclease is capable of cleaving the intron-less copy, and even less the rest of the mechanism. So we really wanted to see that in this model, but unfortunately, the somatic hybrids, always the 3 cases that we observe, retain the intron-containing COX-1 gene from the wild parent, that is, for example, the copy from Hyoscyameae niger or from Hysocyameae, while the copy of tobacco, the intron-less copy, is gone. So we can't see if the intron propagated into the copy and then it was lost or not. And so the intron homing hypothesis could not be tested in these hybrids. And that is all. Thank you very much. This is members of all and current members of my group, the Plant Evolutionary Genomic groups here in Argentina. And much of this work was done with collaborators, Mikhailo Zubko from Manchester Metropolitan University and with Jeff Palmer in Indiana. Thank you very much.
Thank you very much, Virginia. Very interesting the talk and all the work that you have done with your group. So we are now open for questions. If you want to just drop your questions on the chat or just turn on your microphone and you can ask directly.
I have a question. This is Dick Olmstead.
Hi, Dick.
Hi, Virginia. Very nice talk. I really enjoyed that. Thank you. It's good to get caught up on what you're up to. In the case where in the Hyoscyameae Nicotiana hybrid, where the genes sorted out from one or the other parent in most cases, with just a few where both were retained. I have a couple of questions. One is, where they were both retained, were they very divergent so that they were not being recognized as homologous regions? And the second question is, do you think that that's just a random sorting out, or do you think selection is involved?
Okay, so first, the 2 copies that were retained, if we discuss the 3 genomes, there were different genes that were retained in 2 copies. In all cases were the shorter genes. That's one common feature. In all cases, the genes have non-synonymous differences. So if they were important or not, that would be a different question, but at least they were, the proteins will be different, so they should be recognized. And then there's a very good question whether this is a result of genetic drift or natural selection. Maybe both, because the fact that one copy is eliminated in 90% of the cases calls for natural selection. But the fact that together with the genes, the intergenic regions that are also shared between the 2 parents are also lost, then it could be that the genes are taken together with the rest of the intergenic homologous regions, and this is just a subproduct of eliminating homologous sequences as part of the mechanisms of homologous recombination, BIR in particular. So again, I feel that we need to study more hybrids and to understand the process better to respond to that question.
Thanks.
Thank you, Vic and Virginia. Any other question for Virginia? Have you, I know that you have worked for a very long time on Solanaceae, like as a model for this study, but do you know like any other particular group that have these independent gains of these genes in other families?
So are you talking about the COX-1 intron or?
Yes, the COX-1 intron.
Oh, yeah, there are many. So the ones that we studied and found these intrafamilial transfers more frequently would be the rhamnaceae, the apocynaceae. I'm missing one more.
And it's usually more frequent, the gains of these this intron than in Solanaceae, or?
No, even more frequently. I believe that in Solanaceae, so, well, I don't know. So the fact that they sort of grow in the same areas, the Solanaceae that have the intron may explain why those are the Solanaceae with the intron. But no, other families have many more cases of intrahoming.
That's super interesting, thanks. Now, any other question? So here's a question of Federico Roda. I don't know if you want to turn on your microphone, or I can do the question for you. Okay, so his question is the presence absence of the COX-1 intron polymorphic within species?
Well, that we were always interested in finding those plants. In some cases, yes. For example, Ipomoea, we found species with and without the intron. Within a species, so we're interested in both among populations and also within generalists to see the most recent cases of transfer. So Hyoscyameae, I'm pretty sure that not a population study has been done, so it's not really for sure, but the differences in the COX-1 intron in Hyoscyameae, I'm pretty sure that it's very recent, and we will find populations without the intron, for example. Ixora is another genus that has very weird patterns. The fact that we don't have population data, DNA basically, of different species, and from the interesting species data from populations, were not able to study those. But those are my two candidates to do this study.
Nice, thanks. That's very interesting. Yeah. Okay.
I have another question if nobody else does right now. Virginia, in Yuan Yao's work on Hyoscyameae, looking at waxy, he was able to infer that the hexaploid genome of Atropa was probably the result of a tetraploid ancestor crossed with a diploid that is now an extinct lineage that was an early divergent lineage within Hyoscyameae by presence and absence of the transposable elements that he found. Do you have data, do you think, that from the genes that you sequenced for, which is a much larger part of the genome, to look for evidence of that third genome in Atropa?
No, not really. I would like to look at the nuclear genome in this tribe, but no, okay. I don't have any more information on that. I did look at some point, I told you I wanted to look at the retrotransposon in, well, I did look at that now I remember, but no, the results were negative. So yeah, we couldn't learn much about that. Yeah, I remember now. Yeah.
That's interesting, yeah. I don't know, any other question for Virginia? Otherwise, we are finishing the seminar, so thank you.
Federico has one question.
Oh, sorry.
It's there, sorry.
Hi.
Yeah, sorry, Federico, hey.
So from your previous answer, it seems like it's not so common to have polymorphisms in the presence absence of this intron within species, or it's just that you haven't looked enough for variation within species?
Okay, so in general, we think is most of the transfers are sort of recent. So we should be able to find differences, variation at the population level and definitely the species level. We identified in 2008 some clades that the intron acquisition was a little more ancestral, because at least, let's say, 7 genera had the same intron. It was clear that it was an ancestral acquisition. But most cases, we mapped the intron acquisition and it was at the tips. We didn't have that much resolution, let's say, because we analyzed, it was 1,000 angiosperms. So that's not that much. So we saw, we could identify transfers to the tips of that tree that we analyzed. But no, I'm sure there will be variation in at the species level and definitely at the genus level.
So you don't think having different mitochondrial genomes will induce any reproductive isolation between? So that's not the reason why. That will not.
No, exactly. No, I don't see that the intron is affecting in any way to the recipient plant. Apparently, the factors that are involved in the splicing of that intron is nuclear encoded, is like a tRNA synthesase, and probably all plants have it, so that is not a problem either.
Okay, thank you. Thanks a lot.
Okay.
Thank you. Sorry that I almost lost that question. Okay. So thank you, everyone. If there is no other question, now yes, we are finishing the seminar. Thank you very much, Virginia, for this very interesting, very cool talk. Next Friday, we will have the talk of Evelyn Gagnon, that she will talk about the phylogenomics of Solanum and the implications of these phylogenomics of Solanum on evolutionary studies. So see you next Friday. Thank you, everyone. So,
okay. Yep. Bye-bye.